Hello, students. Good morning. Here your teacher Dolcy. And as you all know, we are on chapter number 3rd, Human Reproduction from Unit 1st of your syllabi. Today we are going to start a new topic named fertilization. So, first of all, what the fertilization is. So, fertilization is a kind of process in which fusion of haploid male and female gamete occurs to produce a diploid zygote. So, fertilization is generally known as sungamy because it involves the fusion. Fusion means union of the two haploid gametes that are male and female from two of your parents to produce a single diploid cell that is said to be the zygote. Okay. So next point is what is the site of fertilization and what are the specific regions in the female ovary where the fertilization occur. So in human beings fertilization is internal, it occurs inside the body of the female and it takes place. Actual fusion means actually the sperms are delivered inside the body of the female. After that the fusion of both those gamete it occurs in the ampullary isthmic junction, means where the ampulla and the isthmus, both the regions of fallopian tube they joins, that junction is said to be the ampullary isthmus junction of fallopian tube of human female. At that place the fusion of the male and female gamete occurs. Okay. As you all know that fertilization, the process of fertilization is not too simple. It is quite complex. It comprises number of other further processes that ultimately leads to the fertilization of the female egg with the male egg. So first of those approaches is the approach of sperm to the ovum, means how the sperm it moves towards the ovum to fertilize it. So during copulation, when the male inserts its erectile penis in the vagina of the female, that male releases about 3.5 ml of seminal fluid. Okay, the semen that is about 3.5 ml. This process of release of semen into the vagina of the human female, that process is known as ejaculation or the esamination. And that 3.5 ml of seminal fluid it approximately comprises of 200 to 300 million sperms. So in this small volume large number of sperms are present to fertilize the ova. This ensures the reaching of a number of sperm to the ovum as many as sperms, means from that at least 50% of sperms will be capable of reaching up to the egg in the female fallopian tube, and many sperms that are engulfed by the phagocytosis that occur in the acidic vaginal region. Not all the sperm they reach to the egg, only 50% of those reached. The leftover all are phagocytized by the acidic pH of the vagina. Okay, so only about from those 50%, only 100 of the sperms are capable to survive a long path that of from vagina up to the fallopian tube. Only 100 sperms are capable to reach in the fallopian tube. Okay, the sperms they swim in the seminal fluid with lashing flip flap movements of their flagella with their tail at a rate of the rate. What the speed of the sperm? It is about 1 to 4 per minute only. So speed is not too high, but as we compare the speed with their size, so we can say the speed is quite high. So the sperm when they swim toward the fallopian tube from vagina through uterus, they aided some kind of the aspiratory action that is due to the uterus and the peristaltic movement of the fallopian tube, means to push these sperms toward the ova, some kind of movements that occur in the uterus region, that is said to be the aspiratory action of the uterus. Okay, then when the sperm they get mature inside the female genital tract to fertilize with the ova, that process is known as capacitation. Please take it seriously that what the capacitation is. So the capacitation is the phenomena of physiological maturation of sperms inside the female genital tract to render them competent to fertilize the oocyte. Means some kind of physiological changes they occur inside the ova so that the ova become capable to fertilize the oocyte at a greater extent. Okay, it occurs in the presence of viscous fluid that is secreted by the secretory cell of the oviductal epithelial lines. Some of the secretions that are secreted by the oviduct, they make they make sure the process of capacitation in the human sperm. Okay, and the process of capacitation, it takes about 5 to 6 hours, and the capacitation obviously a kind of change. So the changes that are observed in the sperm during the process of capacitation are as following. So the changes are, first is the loss of decapacitation factor, means the sperm or the factories, they remove all those factor which decapacitate the sperm, means which reduce the capacity of the sperm to fertilize. Secondly, to increase the mobility of the sperm, the calcium influx, it occurs inside the sperm to ensure their mobility. Next is the flagellar movement of the sperm. They change into the whiplash movement so that the movement become rapid. Okay, the last one is the removal of the cholesterol vesicles that are adhering on the surface of the acrosperm so that the acrosperm become free to interact with the cortical granules of the egg and to release the hydrolytic enzymes to dissolve the outermost membrane of the egg so that the sperm nucleus can penetrate inside the nuclei of the human egg. Okay. So, as you all know, that the ova is released from the Graafian follicle of the ovary on the 14th day of menstrual cycle, and that ovulation occurs with the help of the LH surge that occurs on the 14th day of the menstrual cycle. Then the ova is trapped by the fimbriae of the ampulla region of the fallopian tube. Then from that region, ovum, it moves in the tube toward the uterus by peristaltic and ciliary movements that are observed by the muscles of the fallopian tube. At that stage of the ova, it is known as secondary oocyte stage. Okay, at that stage, fertilizability and viability of gametes is limited. Means the viable period of the sperm and ova, it is about just 72 hours after the ovulation and just the ejaculation of the sperm. During those period, both these gametes, they interact, but the interaction is possible only if the male sperm, it swims, because the female gamete, it is non-motile in nature. Okay, so the interaction between both, how this occur, it is by a special type of the theory that is said to be the fertilizing anti-fertilizing type of interaction. It is a very specific type of a species specific type of interaction, means only the sperm and ova of one species are able to interact with each other, not of the different species. To ensure that the fertilizing proteins of one species of the that are present on the surface of the ovum, the fertilizing protein that are secreted by the ovum, when they interact with the anti-fertilizing protein that are secreted by the sperms of the same species. Means if we consider human being, so the amino acid sequence is specific within these fertilizing and anti-fertilizing protein of human being. Only the sperms and ova of same species have the capability to interact with each other, not of different species. This is ensured by the fertilizing and anti-fertilizing interaction between the sperm and the ova of the same species. This is why, because it remove the chances of polyspermy, because only one sperm or ova, they interact in pair to make this anti-fertilizing fertilizing complex, and this way the other factors will not participate in this and the polyspermy will be ignored. Okay, so the next topic is penetration of sperm. So, what is penetration of sperm? It is nothing. When the sperm, it generally comes in contact with the ovum through the animal pole. Okay, the penetration of sperm is a kind of chemical mechanism. It occurs when sperm, it comes in contact with the ovum. As you know, ovum has two pole, animal pole and vegetal pole. I explain you will these with the help of the polar bodies. Okay, so you must know that it is necessary when the sperm reacts with the outermost layer that is known to be the zona pellucida, a kind of reaction it occurs that is said to be the acrosomal reaction. In this acrosomal reaction, the header region carries the acrosome. It releases number of hormones that are said to be the lysine or the sperm lysine. They dissolve the envelope of the egg locally and make a path to penetrate the sperm inside the ova. And this process is said to be the penetration of sperm. These sperm lysines, these proteins are actually acidic in nature. The most important of these sperm lysine, the enzymes which degrade or which break the head of the sperm, they are said to be the hyaluronidase enzyme, which contain the amino acid that is said to be the hyaluronic acid. Hyaluronidase actually it is a polymer of hyaluronic acid which holds the granulosa cells of the chronoradiator together. So it helps in penetration. So it is known as corona penetrating enzyme. And the acrosin or the zona lysine that dissolves the zona pellucida, and it makes sure that only the nucleus of sperm it interact with the nucleus of the sperm. Rest of the piece, middle piece, tail, they all left over outside the ovum nucleus. Okay, so this is a kind of factor which ensures the monospermy, because only one sperm is capable to move inside the ovum. Only the nucleus of one sperm is capable to move inside. So it ensures the monospermy. Okay, so the necessary point that on which we work is the monospermy, that more than one sperm should not fertilize with a single ova until it will cause some kind of problems. Okay. Next point or the next topic that ensures the movement of the sperm, it is cortical reaction. So what is cortical reaction? When the penetration of sperm occur, then a series of processes like the cortical reaction, fusion of gamete, they occurs. So the cortical granules, they appear in the egg cortex region, means the outermost region of the egg, and it occurs once the sperm cell passes through the zona pellucida and the vitelline membrane. It starts lifting from the cortical surface, means it start to remove it. This produces a perivitelline space within the vitelline membrane. So this perivitelline space, it is excluded or removed by the process of exocytosis. Even at some places it remain attached and which become a kind of thickened structure at that place, which is impervious, means it doesn't allow the entry of the other sperm. So in this way, this process also ensures the monospermy and it prevents the polyspermy. So this is said to be a kind of fertilization membrane. If someone asks you what is fertilization membrane, so it is the membrane which is formed by the attachment of the vitelline membrane with the perivitelline space to generate a kind of thickened membrane which restricts the entry of other sperm to the ova, and it ensures the occurrence of monospermy and it restricts completely the polyspermy. So sperm penetration in ovum, it also include some kind of metabolic activities. First one is the separation of charge, that is said to be the depolarization. Due to depolarization, the permeability of the plasma membrane it increase. Due to that, to ensure, as you know, the sperm penetrate, definitely there will be division, so the number of or the rate of synthesis of protein it is increased to ensure the post fertilization divisions. Next is because of rapid movements, rapid divisions, the respiration rate of respiration is also increased, because the metabolic rate at this point is said to be very high, so the respiration rate is observed to be high. So this rate is observed due to increased consumption of the oxygen from the environment. Okay, so cortical reaction is very necessary, because this may not occur, then there will be no physiological changes observed in the ova, so that it finally change up into the fused zygote. For that, a necessary point is to observe the cortical reactions. So the next point of this is, or the last point to ensure the process of fertilization is just the fusion of gametes, both the gamete nuclei, whether male or female. As the entry of the sperm, it stimulates the secondary oocyte to undergo or to continue up its meiotic second division, which produces thus functional ovum and the secondary polar body or the second polar body. Inside the ovum, the sperm penetrate or the sperm nucleus, it take its path, and that path is said to be the copulation path. So please, the copulation path, it occurs after the penetration of the sperm inside the ovum, not at that time. That process or maturation of the sperm is said to be the capacitation. So don't confuse up between the copulation path and the capacitation. It is just the initial step that the sperm take to fertilize with the ova, or to move toward the oocyte stage. But actually the movement of or the path acquired by the sperm to fertilize with the ovum, functional ovum nuclei, that is said to be the copulation path. So the centrioles of the middle piece of the sperm, they form a spindle, because female ova doesn't have any kind of spindle. So these spindle fibers, they ensure the degeneration of the nuclei, fusion of both set of the chromosome. They initially lie on the two poles of the spindle. Later they set the chromosome and mix up the both. That is said to be the amphimixis, because the chromosome are from two different points. So firstly they situate themselves at two separate poles, then with the help of spindles they mix up the chromosomal content, and that is said to be the apomixis. The fertilized egg now, which mix up its genetic material, it is said to be the zygote. And the zygote, it carries two set of chromosome, one from other parent and other of other parent. That is said to be the syncaryon. So this is all about fertilization. Now fertilization, why fertilization is necessary? It is very necessary to ensure the diploid content of the population. It is necessary to avoid or to restrict the polyspermy. It is very necessary because in this process the apomixes occur, which make sure the occurrence of two different set of chromosome parallel to each other, which leads to some kind of variation, and variation are the key factor for the process of evolution. And here the centrioles, they come from the human male sperm, and these centrioles ultimately are necessary for the cleavages that observed in the zygote. And finally, the determination of the sex is only possible because of this movement of chromosome toward the opposite pole. And in this way we can say that the fertilization is very necessary in from every point of view. So after this we will start up with the post fertilization, the cleavage. This is all for today. If you have any doubt, you can ask. Thank you.